Hi everyone. Hopefully you can hear me. Really looking forward to getting to speak to you today. Um, I wanted to start off with a story um, about kind of how I got involved in this. So I'll start my slideshow um, and say that really I really have enjoyed telling this story for several years. It's near and dear to me because it's the first story that when I got involved with online courses several years ago that we taught. And it's also near and dear to me because I took a high school biology course. And when I walked out of that high school biology course, I vowed to myself that I would never come back to biology, that biology was largely just memorization, and occasionally they might let you cut up a frog. But that largely, it was something that I just wasn't interested in. And what I love about this particular story is it shows how starkly different some of the research in modern biology really is, especially once we start using computation. And so as a result, it's a very short on-ramp to showing someone how really interesting it can be to apply what we'll see is some relatively simple analysis to answer a question that would take us hundreds of thousands of dollars to do for thousands of bacteria probably, but that with a computer can be solved in a matter of seconds. And so I hope I've at least intrigued you. And what I'll do in the following lecture is demonstrate how we can use programming to actually solve the problems that we'll encounter today. But I like separating out this material and discussing it on kind of a theoretical level. And that material is finding replication origins in bacterial genomes. So I need to give a very short introduction to the science that we would do for anyone who hasn't seen it. And where it begins is at what you might think would be, or what some people would think would be really the start of biology as a computational discipline. Because once we start to unlock the identity of molecular biological molecules, like DNA, we realize that we can start analyzing something like DNA as a strictly defined data set. So I'll explain kind of what I mean by that shortly. And that the writing is on the wall that biology will eventually be a computational field in 1953, when we have a double helix structure of DNA and when we know that it consists of two opposing strands of ACs, Gs, and Ts. There's one quote from the Watson-Crick paper in 1953, though, that I would focus on first, where they say that it has not escaped their notice that by having a double helical structure, there would immediately be a suggested copying mechanism for the genetic material, i.e. DNA. And what they mean by that is that you could have two strands of DNA, so you have complementary strands, A binding with T and G binding with C. You could imagine that these strands live in kind of a soup of nucleotides, so you have free-floating nucleotides that can be used to when the strands are unzipped, essentially go in and bind again. So we unzip a double-stranded DNA molecule, and then we provide free-floating nucleotides to produce two copies of our original DNA, where each copy kind of has used these free-floating nucleotides for one of its strands. And so you might pick out which of these three hypotheses was what Watson and Crick hypothesized. And I can run through that, kind of at this time, there were three immediate hypotheses for what the mechanism for how DNA replication might operate. You could have on the left a semi-conservative model where after one round of replication, each of your double-stranded molecules would contain one strand of your original DNA, which here is shown in orange. And then after two rounds, you would have four molecules, and two of them would have DNA that was not present in your original generation. Or you could have a conservative model where, however the DNA replication process is happening, you wind up with two molecules where one of the molecules has 100% of the parent DNA, and then the daughter DNA makes up of the other molecule. And then you could have a dispersive model which proposed that maybe this process kind of jumbles up the DNA. So it gets swapped across or something throughout this process. You have a uniform, almost, distribution of parent and daughter DNA across all your 4 strands. So each strand does not come from either the daughter or the parent, but is a mismatch of new DNA and parental DNA. Okay. And so my question would be, which of these was Watson and Crick's? And we can see that, well, their model was saying that after one round of replication, you would have 2 double-stranded DNA molecules, and each one would have 1 parent strand from the original molecule. And so you have a semi-conservative hypothesis. Now, that has turned out to have been confirmed, or I should say it's never been disproven. But the question is how we would even demonstrate something like that, because DNA is something that's submicroscopic, if you want to think about actually reading out the letters of A's, C's, G's, and T's. And it's very difficult in the 50s in order to get at this. It took decades in order to find a technology that could actually read the A's, C's, G's, and T's. So we see a general theme of biological and, in a larger sense, scientific research, which is that we have to come at our answers very indirectly often, given the tools that we have to solve the problem. And so Meselson and Stahl, 5 years later, produced what has been called the most beautiful experiment in biology. And many people have seen it, but my experience is that many yet still haven't. And it's a valuable experiment just to see for the beauty of it. So their insight relied on the fact that one isotope of nitrogen, so nitrogen-14, is lighter and abundant than the nitrogen-15, which has 1 additional neutron. So for that reason, bacteria or any organism is going to have greater access to nitrogen-14 in nature. And so what they did was they placed E. coli into a 15N medium for many rounds of replication, and that caused the bacteria, in a sense, to kind of pig out and gain weight as they absorbed this heavier isotope into their DNA. And then what they saw was, well, let's transfer that heavy E. coli back into a more natural, less dense 14N medium. And so the question becomes, well, what's going to happen when they do that? And the insight here is that any of that daughter DNA that's produced when you take the fatter, in a sense, E. coli that's had the heavier nitrogen, any daughter DNA that will then be produced will use the nitrogen that's available in the less dense 14N medium. So any of the daughter DNA produced would be lighter. And they used this insight to say, well, let's spin the DNA after 1 round of replication. We can measure the replication time for E. coli. And after this has happened 1 round of replication, we're going to spin the DNA in a centrifuge. And the question that I always ask students is, why would they have done that? Why would spinning the DNA in a centrifuge have produced a potential answer to their question? So it's a good time to ponder that and look at the 3 different hypotheses that we have. And you'll notice that if you look at the semi-conservative model on the left, you'll have, after 1 round of replication, that each molecule of DNA will have 50% parental DNA and 50% daughter DNA, and so you won't have any difference. Every molecule is going to be essentially have the same weight. Okay. Whereas if we look at a chromosome in the conservative model were to be true, then we would actually see a separation. We would be able to differentiate by the mass the older DNA from the daughter DNA. And if we think about the dispersive DNA, well, you're going to have a similar story to the semi-conservative model because everything has approximately the same, every molecule has about the same daughter DNA as it will have parental DNA. And so this will allow them to see what happens, and based off of what actually happens, maybe rule out one of the models. And so what they found was, indeed, you couldn't you did not see any differentiation between the DNA. Everything appeared to have the same mass, so when they spun the DNA, they didn't see a division. And then the question becomes, okay, well, they've immediately, through this very elegant experiment, eliminated the conservative model, at least for the E. coli that they were working with. It could be that different life forms, prokaryotes and eukaryotes, or different types of bacteria even, operate on a different model, but they at least were able to eliminate the conservative model for what they were working with. And then they decide, well, what would we really see if we do this for another round of replication? So we take the same DNA, they're still living in the lighter medium, the 14N medium, and we're now going to spin the centrifuge again after 2 rounds. They say, okay, well, what's going to happen now? And you'll notice that whereas after 1 round of replication, you couldn't differentiate DNA in either the semi-conservative or the dispersive model, the story is different after 2 rounds of replication. So after 2 rounds of replication, we still would see a uniform distribution of the masses of the bacteria. So we would see just a big clump of DNA if the dispersive model were going to be true. But for the semi-conservative model, some of those molecules, you have 4 of them, half of them will have only 14N, and the other half will have 50% 14N and 50% 15N. So those you will see that half of your DNA is heavier and half of it is lighter. And so when they spin the centrifuge after 2 rounds of replication, what they see is indeed you have a kind of 2 groups of DNA. You have heavier DNA and you have lighter DNA. And so they had eliminated the dispersive model, again, at least for what they were researching. And so because it divided into 2 different densities, they provided very strong evidence for the semi-conservative model. And I always ask students to stop here and say, does this prove that the semi-conservative method or hypothesis must be true? And we'd say, certainly not. That we've only found that the semi-conservative hypothesis has evidence for it under the conditions of the experiment that we've run. However, this hypothesis has stood strong as a rare example in biology of something that appears to be pretty universal, that all organisms replicate their DNA but based off of the semi-conservative model. So it's a very powerful experiment, but also a very simple one, which is why it's called the most beautiful experiment in biology. At any rate, when a biologist looks at this process of a bacterium or a cell replicating its DNA, they see all kinds of complex processes through the cell cycle that are required to orchestrate this symphony of actions that will cause a bacterial cell or any cell to be able to replicate all of its DNA and produce 2 cells from 1. When a computer scientist who was a biology phobe like I once was looks at a problem like this, they say, well, actually I have a string of DNA, which is just a contiguous collection of symbols. And if you followed along with the programming, we know that that's a built-in type in many languages, where we'll have access to work with strings. And it's going to motivate what we do next. But for now, I'll just say a DNA string is a string of A's, C's, G's, and T's. And because of the complementarity, because A always binds with T and C always binds with G, even though DNA is double-stranded, you get that opposing strand, that complementary strand, for free. So with the DNA string comes the complementary strand, and so the computer scientist reasons that I only really need one of those 2 strands, and so I can just think of a genome or an organism's chromosomal DNA as just a long string, and yep, there's this complicated biological process that I'm sure is very involved, but at the end I wind up with 2 copies. So I have 2 copies of my original string, and there's nothing of real computational interest in this problem. So let me back up from this and say, well, I bet we can find some. And the way we're going to find some is to say, well, there's more to this than meets the eye, and yes, we could get very complicated in terms of what's going on inside the cell. But for now, we'll say replication has to begin somewhere. And we could call that the replication origin, which you could denote by ORI. And so in a bacterial chromosome, you will see a single replication origin, and then based off the semi-conservative model, you see kind of in both directions around the circular chromosome, new DNA is added in, so we wind up with 2 circular chromosomes. In organisms like ourselves, so in more complex organisms, you'll see often multiple linear chromosomes, and within those chromosomes you have multiple replication origins as well. So you have more DNA typically to replicate. Something like E. coli may only have approximately 3 million letters in its single chromosome, whereas you or I will have 3 billion letters, so it's divided across chromosomes. You have multiple replication origins. This is much more complicated, but for something like E. coli, a simple bacterium, then you have one replication origin. And there's a natural question there, which is, okay, where is it? This is a very natural scientific question to ask, especially as we start thinking about engineering bacteria or so on. If we edit a bacterium, we don't want to change its replication origin because that will disrupt the replication process. So I could state this as something of interest. I'm going to give you a DNA string as an input and call this origin of replication problem or finding ORI problem. And I want you to write an algorithm, hypothetically, that's going to find the location of ORI in the genome. And again, I ask that we step back from this for a second and say, is this a computational problem? And to recall what we defined as a computational problem, which is that we have very well specified input as well as well specified output that uses the input in some way and that produces a solution for that input that can only be interpreted in one way. So last time we saw a GCD problem where I give two integers as input, say two positive integers, and we produce the greatest common divisor of those integers as output. So the natural question then is, this seems reasonable as a scientific problem. Here's a DNA string, what's its location of the replication origin? And we ask, is that a computational problem? And many students will immediately answer yes. But the idea of finding the location of the replication origin is still not perfectly well defined. Do I mean an exact location? Does it happen between two nucleotides? Are we looking for a range in which it occurs? So even something like this that scientifically is clear, computationally is not. And often what we see in computational biology, but many other disciplines, is that there is an art to formulating a problem computationally so that we can start writing algorithms for it. Because a computer will not, if there's multiple ways of interpreting this, we're not going to be as rigorous as we could be. So if we were to present this to laboratory biologists, at least traditionally, and say, how would you find the replication origin? They'd think about experiments like knockout experiments that would eliminate different regions of DNA through the genome. And then we would ask, for each of those things that we knocked out, is it possible for the genome to replicate without each of the things we knocked out? And we would at least have be on the right track of knowing where to look if we knocked a region out of the genome and we wound up with something that could not replicate. If we give this to someone who hasn't had any biological background and wouldn't be comfortable in a lab, but is strong computationally, an ideally strong computational person who hasn't done biology in 20 years would then say, I still need more information. Okay, so we need more information in order to hack this problem. I use the same term hack there because computer science is still very much a science. And often in science we work by trying to break something or mess around, play around with some system and see kind of what results when we do it. So let me give you an experimentally verified replication origin, and let's use that to see if we can reverse engineer some process to find something computationally. And a whole separate discussion here is how in the world we know a replication, or not the replication origin, but an entire genome of a bacterium. Even a bacterium which has several million nucleotides, each nucleotide is smaller than what we can see with a traditional microscope. And this has been an entire research field that's a landmark area of biology that allows us to read DNA and determine what the blueprint is of a living thing. For one human genome, we spent $3 billion in the United States and 10 years in the 1990s just to get a single genome. And through this process, we saw in kind of the mid to late 90s a lot of bacteria being sequenced, and this has brought on a boom of sequencing of all different kinds of organisms. And so that is a separate discussion in and of itself that I also love having. But for now, I'll take for granted that I can go to an NCBI database and download all kinds of different DNA blueprints for any organism I like. And here's one that is the bacterium that causes cholera, Vibrio cholerae. And here's its verified replication origin. And it's a window of about 500 nucleotides long. And there must be some property of that window of the DNA that makes it special, because if DNA truly is this blueprint for everything that goes on in the cell, something, for example, a protein may come in and bind to some region in that DNA that's going to actually start its replication there. There's no additional information in this system. It's just the sequence of letters that can be viewed from outside. So there's got to be some hidden message that tells some process in the cell to initiate replication there. And so now we're getting closer to something that's well-defined computationally, and we would ask the same question. And so here our computational problem would be, say I give you not the whole genome this time, but I give you a short string that I call text that represents this window of the replication origin, and I want you to tell me a hidden message that you find. And again, we ask the same question. Does it represent a computational problem? And if we use the same analysis, we say the input for this problem, much like before, is well defined. The input is a string, fine. But the output here, a hidden message in some text, is not defined in a way that we can only interpret it in one way. What does hidden message mean? Does it mean something that has a lot of T's in it? Does it mean something that occurs infrequently? Does it mean something that occurs frequently? There are many different ways that we could interpret this. And so there's two scientific problems here. One is if I give you an entire genome, say approximately 3 or on the order of 3 million base pairs, a few million base pairs long, where is the replication origin? Where is the window that it starts replicating its origin? And again, I'm saying these are scientific problems. I want to define them formally. And given the replication origin, so given a short window, what is that hidden message that indicates to the cell that replication should start there? So those two questions are going to drive what we do. And I want to formulate them precisely computationally. And eventually we'll come back and program them, but I want to demonstrate how we'll be able to solve them now. And then I want to kind of, in a later lecture, step backwards from this, program the algorithms that we produce, and see if we can produce the results that I'm going to show. So I'll come back to this first problem of one of the problems, which is finding hidden messages in the replication origin. If I assume I'm given wherever ORI is, and I think of that as a text, can I find a hidden message in it? And of course, that's not defined. It's not defined in a way that I can only interpret it in one way. Like for a GCD, I can only interpret that in one way. I can only interpret yes or no of whether an integer is prime in one way. But I can interpret hidden message however I like. And so if we think of that we need a bit more biology, we could learn that in this case, you have a special protein called DNA-A that initiates replication. And this is a complicated internal process, but on the basic level, this protein DNA-A binds to a short segment in the replication origin that's called a DNA-A box. So that's our hidden message of sorts that says bind here. And the question is, what is it that makes that region special? And so we think, well, here's an important biological question. If we're reasoning about what this DNA-A box might look like, would it make sense if I'm an organism for me to have multiple DNA-A boxes, or would it make sense for me to just have one? So I may stop, at least for a moment, and let you think about that. And then I'll discuss kind of what the answer is that we'll see. So I sometimes tell my students that often in biology you have very complicated processes, but the answer can sometimes be reductively easy, and that you can answer a surprising number of questions in biology with a one-word answer. And that's the one-word answer that Theodosius Dobzhansky gave us, which is evolution. So here, if I have multiple DNA boxes, if there's some process that's causing mutations, there is less of a chance of that mutation disrupting my process because I have multiple boxes, so I could afford to lose one perhaps, as well as just the fact that if this is related to protein binding, there's a higher probability of the binding happening if there's multiple locations for it. And so those two things are hand in hand, and we have that organism with multiple DNA boxes would have higher fitness. So Dobzhansky says nothing in biology makes sense except in the light of evolution. And so we can reason through why we might see this. And in fact, it is true that we will see this, but it will put us on the right track of saying, well, if we reason through that probably organisms would have multiple of these boxes in order to be more fit, that's going to motivate what we do for finding frequent words. So what we're thinking about looking for then is surprisingly frequent substrings. We'd say there's been an experimental process. We're really glad that it has produced a verified replication origin for us in this bacterium. And now let's look through it and see if we can figure out if there's something that's surprisingly frequent. And that's going to motivate the problem of counting how often a given substring occurs. So what's nice about this is that this is one of the simplest string problems that you could possibly formulate, which is I'm looking for a contiguous string or a substring within a longer string. So it's a pattern matching problem of finding how many times a given pattern occurs in a longer text. I could call that substring counting or counting words. So we have a string pattern and a longer string text, and I want you to tell me the number of times that it occurs in text. And we say, okay, great. It's a perfectly well defined computational problem. There's only one way of interpreting something like this. How many times does a word occur in a longer text? And yet, we could think about, well, here's an example. How many times does ATA occur in this longer string? CG, AT, ATA, TCC, ATAG. And I will often poll my students for a question like this and ask them, and half the class will vote that it should be 2, and half the class will vote that it should be 3. And the answer is that it really could be either. So you may have a built-in package, for example, that chooses one of these for a programming language. And for this particular application, we'll go with 3. You could make the argument that it shouldn't be either 2 or 3, because for the overlapping ATA, that middle A could get mutated away and you would lose 2 potential binding sites as opposed to just 1, in the simplest way of reasoning about it, assuming that, for example, one single nucleotide mutation would disrupt the process. And so you could say, well, if that's the case and a single mutation can throw out multiple of these words, maybe it should be something like 2.5. Okay, so there's even more than 2 possible answers to this. But for this application, 3 is an integer value, and it makes more sense than 2 because there will be multiple locations of binding. So we will count overlaps. And there's a key point here, which is that how I'm going to think of a string is just as an array of symbols, at least for now. And so the string itself, if it's an array, let's think of it as being 0 indexed. We have some text. Here's a text, and I have, it has length 14. So I'll think of the initial symbol as being index 0, and that will range all the way up to index 13. And the notation that I'm going to use for this given substring, so a substring of the text, meaning a string that's contiguous within the text that I've highlighted here in pink, is text of 710. And that will always strike people who haven't seen this notation as odd. I can see why we would call it text of 7 something, but why not text of 7.9? It starts at position 7, according to the zero-based indexing, and then it proceeds up to and including position 9. And so that always will seem weird to students, and for good reason. It doesn't kind of jive with how we think about counting. And so, as a quick quiz, you could stop and say, how would we refer to this substring? And so we say, well, it's the substring that starts at position 0, and it goes up to and includes position 2. So we would say, well, we want under this notation, we'll say text of 0, 3. And I could have you pause again and think, what about this substring? And then we say, well, what's highlighted starts at position 3. It goes up to and includes position 5 and comes short of position 6. So we'd say text of 3, 6. And there is a pattern here. There is something that we can notice about the way that we do this indexing, and it's going to be the reason why we do it, or one of the multiple reasons why, that makes it convenient to work with. And for that reason, many modern languages have kind of transitioned to this way of notating substrings, or indexing substrings. And if you think about what it is that we notice here, well, it's that in all of these cases, you easily get the length, the length 3 substring every time, and you can just subtract the lower index from the upper index. So here, this is text of 3, 6, and you get 6 minus 3 is 3. Before we had text of 0, 2, well, or 0, 3, excuse me, even though it only included positions 0, 1, and 2, but you can quickly subtract 3 minus 0, 3. We had text of 6 to 9, or 7 to 10, excuse me, where we had TCC, that was a length 3 substring, that started at position 7, went up to and not including position 10. We can quickly get that it has length 3. So under this, how would we refer to a substring of a text where the substring has to have length k, and we want the substring to start at position I? So I could ask you to think, well, generalizing that notation, what does that mean? It starts at position I and it has length k. And we realize, well, the difference between the upper index and the lower index has to be k. We know that the lower index is I because that's where it starts. And our answer then is text of I to I plus k. And that's very, very useful because we can very quickly see the length of the substring by subtracting the top from the bottom and seeing that that's just k. And those are really the two pieces of information that we care about often in applications, which is where does the substring start, what is its length? And if I've got those two things, then if I've got the starting position and then whatever is past the ending position, I can get the length for free. So it's a nice notation. I will note that we have worked with arrays already, but I haven't talked about subarrays. So I haven't talked about the idea that we could have, for example, an array of factorials, or an array of prime Boolean values where it corresponds to whether a given integer p is prime or composite. So we'd have true or false. You could imagine if I'm only interested in the primality of the integers between 100 and 200 for whatever reason, we might be interested in that subarray that goes from 100 to 200. And so we'll use that same notation. So if something went from 100 to 200 inclusively, we would denote it as 100 to 201. Okay, and so we'll come back to that when we program things with respect to this material. But for now, I'll just point out we can already think algorithmically about how to count patterns. We can already move toward writing the algorithm that we produce in pseudocode. And so here's our idea for counting patterns. I can imagine sliding this window, for example, of length k, here k is equal to 3, along the text and saying, where is it that I see a match? So if my pattern is ATA, I could start at text of 0 to 2, and then text of 1, 3, or 0 to 3, excuse me, text of 1, 4, text of 2, 5, and so on, and compare each of those little windows of the text against a pattern. And I would start adding 1. So I would increment some count variable behind the scenes every time that I see a match in that window. Okay, and so it'll actually be vital to know how many of those windows I need to consider, because this is going to require us to write a loop. So we'll have a for loop that's going to allow us to do multiple windows. And that for loop is going to start at I equals 0, and it's going to need to know how many of these windows I need to do. In other words, how many substrings of length, for this example, k equal to 3 are there in a string of length 14? The more general question is how many substrings of length k are there in a longer string of length n? So that's another place where I'm going to pause and ask you to think about that for a second, because it will come up repeatedly, not just in biological applications, but it's a good exercise to quickly know the answer to in all of string algorithms. So I'll pause for a second. So for this example, you have n is equal to 14, k is equal to 3, and you can count and see that, well, there are actually 12 different windows here. And you can, if you shortened k by 1, then you'd get a 13th window. If you lengthened k by 1, then you would lose one of your windows, because you don't want to consider something that goes off the end of the string. If you lengthened n while keeping k the same, then you would pick up an additional substring if k were the same. And so you have something that's with respect to n that's also, when k gets bigger, every time k gets bigger by 1, the number of substrings decreases by 1. So you have a simple linear relationship here, and you start to see, it's n minus k plus 1. Okay? So the longer n is, the more of those substrings you're going to have by 1 each time. The longer k is, you're going to have 1 fewer each time. And then you have a plus 1 because it's a fence post problem. So you'll see that for this one, you have a length 14 string and k equals to 3, but there's not 11, there's 12, because you have 12 possible starting positions. Okay. So I'll pause for one additional exercise here while we're at it, and say that really you have enough already, even with the basics of pseudocode of what we've done in the initial part of this course, to write a pseudocode algorithm that would count occurrences of a pattern in a text. So we're going to have a function, we could call it count patterns or pattern count, that will take a pattern and take a text and produce the number of times the pattern occurs in text. So I'll pause for a couple of minutes this time, and let you at least think about what the pseudocode for that function might look like. every language will have a function that's built in that will tell us how long a string is. So the first thing that we would do would be to access the lengths of pattern in text. So len might be the function, but it could be called something different in different languages. And so using this idea, we have a count variable that needs to start at 0 since we've not found anything. We're going to need to range over the string and check whether a given pattern is equal to a text. And every time we find something that's equal, we're going to update count by adding 1 to it. So we set k n equal to the length of pattern in text repeatedly. And then we have a for loop, and here we needed to know how many substrings to continue. So I ranges from 0 up to, and here we think, well, it needs to include n minus k plus 1 starting positions. So it goes from 0 up to n minus k. Okay, so just as between 0 and 10, there are 11 numbers if you include 0 and 10, or 11 integers. There are n minus k plus 1 integers between 0 and n minus k. So we're going to, that's our range. I starts at 0, it goes to up to n including n minus k, or up to but not including n minus k plus 1. And for each of these i's, we check, is text of I plus k? That's that substring of I of length k that we saw before. Is that equal to pattern? Which means if we line up all the symbols, are they equal? That's another thing that will be built in, typically. And so if we see equality there as strings, then we're going to update count by adding 1 to it. And then after this for loop ends, count will store the number of matches that we found. Okay. So the idea of a string of length k is fundamental to computational biology and gets the term k-mer. And so we say that a k-mer, or a string of length k, is a most frequent k-mer in a string if you don't see any other k-mers that occur more frequently than that pattern does. And reflecting on the fact that we were looking for, at least according to Dobzhansky's reasoning, something that would be frequent. If I give you a string text and an integer k, and I might ask you to give all the most frequent k-mers in the text, because perhaps you could have a tie. 2 formers occur 8 times, and that's the maximum. Now we would say, is this problem clearly stated? And we ask ourselves, well, is the input well formulated? Text and k, well formulated as a string and an integer. Is the output well formulated? Is there any way that I can interpret this desired output as taking the counts, or examining the counts of every k-mer that occurs in the text and selecting the k-mers that have the maximum counts? And there's no other way of interpreting that, so we're at a problem that's well formulated as a computational problem. And once we have a solution or an algorithm to this that will solve this problem, we want to reflect on this experimentally verified replication origin and apply our approach to it. So here's an example for what I was indicating. If you have this hypothetical text and k hypothetically equal to 3, then your most frequent words in this case would be ACG and TTT. They each occur 3 times. TTT overlaps in one of those places, but if we count overlaps, we get 3 times for each. Now I'd ask you to pause for a moment and tell me, if we were interested in solving this problem with an array, how might we solve it? So we already have worked with arrays in the past. Is there any way that we could use an array to help us out to solve this problem? So is there an algorithm using an array that could eventually produce the most frequent k-mers in a text, given that string text along with an integer k? And when you're thinking about this, we don't need necessarily pseudocode at this point. We could just think about planning the algorithm just like we planned it before. Well, I'm going to slide a window and I'm going to keep adding 1 to count every time that I see a match. So I'll have you pause just for a bit and think about how we could solve this problem with an array, as well as what we might find useful as a subroutine. Here's one hypothetical solution. I'll often have many different solutions that students produce. Everything from trying to consider possibly all strings of a given length and counting how many they each occur and then taking the maximum. That one might be tricky here if k is big, because there will actually be 4 to the k different substrings or different DNA strings of length k. So if k grows, that becomes a very big number. This is a candidate solution that we will see is actually by far not the best solution for solving this problem, or the fastest, but it's one that will work. So I could create a count array of length of text minus k plus 1. So each element of the array is going to correspond to a starting position in the string. And then for each I, I'm going to set count of I equal to the number of times that the substring of text, starting at position I of length k, so text of I at I plus k, appears in my text. And then I'm going to say, well, what has the maximum value of count of I? And I could show this. It's easiest to show with an example. So if text is this longer string and k equal to 3, text of 0 to 3 is just ACG. And I would say, how many times does it occur in my string? It occurs 3 times. How many times does the substring of length 3 starting in position 1 occur? It occurs twice. And I could iterate through the string. So I could use the function that I've already written to help me as a subroutine, because I already wrote something that will count how many times a substring occurs in a text. So here I hit TTT, here I hit TTC, I use my subroutine, and I fill in the array as we go, each time finding the number of occurrences of the substring of length k, starting at position I, and filling that as count of I. Okay? And at the end we see, well, what occurs the maximum number of times? The max value is 3, and the strings that correspond to that are going to be the strings at position 0, 3, 7, 10, 14, and did I miss anything? 11, 10, 11, and 14. So that seems like it's going to be 6 strings, but it's actually just the strings ACG and TTT. So we're overcounting each of these by 3 because they occur in 3 places. So here's a lengthy function, not that lengthy, but a function that would do this. And I could walk through it and say, well, what are the subroutines that are going to be needed? I've colored them here. It's a little longer than some of the functions that we've worked with thus far, but the ideas here are no more complicated. So I could start with by setting a frequent patterns array of strings. And so that array of strings, if you think of kind of an array as a list, it will essentially just form a list of strings at the end, and that will be our frequent patterns. So at the very bottom, I'm going to return that. In the meantime, I'm going to say, well, set n equal to the length of text, form my count array to have be an array of integers of length n minus k plus 1, have this for loop that will range up and set the values of count. So for every integer between 0 and n minus k, set count of I equal to the pattern count of how many times that substring of text of length k starting at position I occurs in the longer text. And so that fills that count array that we saw before. Then it's just a matter of taking the maximum value in that count array. So I need a subroutine that can find the max value in an array. That may be built into a language, or we could write it. It's just a matter of ranging through an array. And then we say for every integer I between 0 and n minus k, we're going to go in and say, is it the case that something achieves the maximum? And if so, I'm going to append it to the list that I've already produced. So I'm going to need this append subroutine that will take a list as well as some new pattern and simply add that new pattern on to the end of the list. And at the end of this, if something has occurred m times, for example, I've added that pattern m different times because we saw before that we had strings that occurred 3 times, and we would have added each of them 3 times. So I'm going to throw out the duplicates at the end. I'm going to need some function that takes a list of patterns or an array of patterns and throws out any duplicates that we have. And then I'm going to have my collection of patterns. Now, then we could write each of these subroutines, but I just wanted to point out that we can at least plan what it is that we needed to do without writing these subroutines and thinking about these as separate functions, which is going to make our work with programming much easier. This type way of programming where we're not trying to do everything at once, but we're using subroutines wherever possible, using modularity, meaning that each function kind of serves one small purpose, one small bite-sized purpose, will help us reason through problems and how to solve them in a context that's more general than any particular language. We can write subroutines in any language that we like, but if we think through problems this way, they'll be easier for us to process as opposed to doing it all at once. And I always ask students, so I typically will have students who are curious, but isn't this a somewhat inefficient approach? And you could think about, if you wanted to pause for a moment and think about why this algorithm is inefficient, or maybe even how we could make it better if you were particularly precocious, and why that might be. And in this case, I would say we have an algorithm that's not as efficient as it could be. For one, you'll have noticed that we keep adding all these duplicates and then we throw them out at the end. Wouldn't it be nice if we remove the duplicates first? So I saw that comment, or not adding something if it's a duplicate. It seems like a silly thing to do. Another thing is a deeper insight, and that would be to realize that when we form, every time that we set a value of count of I, every time we count, how many times does this pattern occur in the text? Every single time we do that, we call this subroutine, and that subroutine scans through the whole text. Wouldn't it be a whole lot simpler to make one single scan along the text and count and have a table, for example, that says, if I see an AAA, I'm just going to add 1 to whatever the value of AAA is. And the next one might be AAG. And I'm going to add 1 to whatever the value of AAG is, and so on. If that's how we were doing this, ignore the computer for a second. But if that's how we were solving this problem on pencil and paper, it's how we would do it. We would form a table, and every time we saw a new substring, we would create a new entry in the table. Every time we saw something make its value 1, every time we saw something that we'd seen before, we would just add 1 to its value. And at the end of that process, we would have a table containing how many times each substring occurs, Kmer substring occurs in the text, and we would just take whatever has the maximum. And so this pseudocode function that I provided does not do that. And sometimes if we think about how we might do something on pencil and paper, that can elucidate how we can get a faster approach. In this case, it's going to provide a faster approach that only makes one scan. And that's our faster frequent words approach. And there's lots to do with respect to strings, syntax, in whatever language we're working with, or working more with arrays. So how will we append to the end of a list? How do we remove duplicates function? There's lots to do currently, but I want to use the benefit of pseudocode and reasoning about algorithms much more generally than any specific language to cover an additional data structure that we'll often work with. And that is to say that when we've worked with arrays or slices in Go or another language, then we think of arrays as lists of variables. So a string, hi there, might be H, I, space symbol, T, H, and so on. You could have an array of Fibonacci numbers, but the indices are always going to be integers. So if I have a list of strings, the indices will always be integers for an array. But what if the indices aren't integers? What if I wanted to produce my table where I'm scanning down a text and I've got the patterns that I've seen thus far, and every time that I see a pattern, I increase its count value by 1. Something like this where the indices aren't integers. The indices here are the substrings, A, AC, and so on. Those are the indices of my array. That would make things much easier when finding frequent words. And what the nice thing about many modern programming languages is that they have built-in maps or dictionary variable types that will allow us to generalize the idea of an array to keys that aren't necessarily integers, or that could be integers but that aren't necessarily 0, 1, 2, 3, 4 up to the length of the array minus 1. And so in this more general version of an array, you have keys, and each key has a value that it's associated with. So here the value associated with GA is 23, which we interpret to mean as maybe that's the number of occurrences of GA in some string. And just as we use a variable name to refer to an array, we'll use a variable, say FREQ or whatever we like, DICT, to refer to a map. All right, and accessing the values for the value of a given key works the exact same as it should for accessing the value for a given index of an array. So if I want to access how many times does GT occur in FREQ, I would just say FREQ of the string GT, and that would tell me 30. Note here that not every string of length 2 is a key. So we don't have to generate all the substrings of length 2. We just have to consider the ones that would occur in the string. Or for a different example, we could just have any set of strings that we like. And so I could rewrite my frequent words pseudocode here that will eliminate subroutines. We won't need to remove any duplicates. I'm not going to need to appeal to this count function that we had written before, and it's going to be much faster. I could have you implement each of these approaches when we learn a little bit more about how to implement maps as well as work with strings. But a better approach would be to say, well, we could have an empty array of patterns. That's eventually what we're going to return at the bottom. And we'll set n equal to the length of text, and we'll do this ranging. And now we need this empty map called FREQ map or FREQ. And I'm going to say, let's do this ranging. And what's my current pattern? Well, it's text of I to I plus k. And that substring of text of length k starting at position I is my current pattern. And I'm going to check, does that exist in my table? If it doesn't exist in my table, I'm going to create a new value for it of 1 associated with that key. Otherwise, if it is already there, say freq map of ATG is equal to 4, I'm just going to increase 1 of that current value. And then that's going to produce this map that will count all the occurrences of each K-mer substring that occurs there. And I'm going to need a function that finds a max value in a frequency map, or in a map of strings to integers. So what's the biggest integer that I see? It's 86, so that must be the most frequent K-mer. So I'll call that variable max count, and then I'm going to just range through my table. So what are over all the keys in my table? If frequency map of pattern equals that max, I'm going to then append whatever that string is to my list frequent patterns that I started with up at the top of the pseudocode. So at the end of that process, I won't have any duplicates because I'm only adding a pattern that occurs the max number of count times one time, because it only occurs in the table one time. And at the end, I'll return the frequent patterns. And I've only made one scan down text, as you can see. I only have one for loop there as I ranges from 0 to n minus k. And I'm just checking whether or not it exists in the map. So it's a much quicker algorithm as would allow you to verify. I'm going to also point out that all this code in red could be written as a subroutine, and if this seems like a tricky algorithm now, it will become simpler when we write this code in red as a subroutine, when we realize that that code in red is just producing a frequency map. All it does is, for text and k, produce the table that we wanted, where all the substrings of length k are the keys, and the values are the number of times that they occur. And so if I look at my better frequent words function now, all it does is form an empty array of strings. That's going to be the frequent patterns. It builds the frequency map. So I'm passing this detail to a subroutine and say, the subroutine is going to build this table for us. And then what's the max value in the table? Okay, that'll be a separate subroutine. And then I'll say, okay, I've found my frequency map. I've found the maximum. What in that frequency map? I'm going to range over all the keys, all the strings pattern, and say if something achieves the maximum, I'm going to append it to my list, and I'm done. And then I've passed all those details to a subroutine called frequency map. That's a separate function. Its input is a text and an integer k, and it produces this chart, this frequency map, which I walked through before. I've not changed that code any in moving it into a subroutine, but it makes the higher level function, better frequent words, so easy to read and understand, I think. So we're going to say, well, let's apply this approach back to the replication origin of what we know is an experimentally verified replication origin. And here's our replication origin, and let's apply an approach that we produce. I'm going to assume that later we're going to write some code and solve the frequent words problem. And we're not going to necessarily know what value of k to use right off the bat. A priori, it could be anything over a range of values of k. And we could form a table even where we say, for a given value of k, what are the k-mers that occur most frequently and how many times do they occur? That'll give us some useful information and be able to start analyzing this and start seeing patterns here. So one thing that you might notice is that when you go between, say, a 6-mer and a 7-mer and an 8-mer, the most frequent words correspond to just tacking on a symbol each time. But the count also goes down. So there could be some noise there. It could be that when we go from a 6-mer to a 7-mer, and we just appended an A onto the start, that A might be irrelevant. It could just be that we got lucky that 5 of the 8 times that we saw an A. But then when we get a little bit longer, then we see that actually we have a string of length 9 that occurs 3 times in this short window, and we actually have 4 of them. And when I highlight them, you'll see that there's even some overlap with those. And I always ask students to say, does this help us see kind of any pattern here that might be existing among the different 9-mers that will actually help us realize that k equals 9 is a very interesting value to use. So especially if you have a background in biology, you'll maybe see this quicker than others, as well as if you might think about why I've colored the strings in this way. I'll continue on for now, though, and say that one more biological fact that I've already mentioned is that DNA is this double-stranded molecule. In this case, on the biochemical level, the 2 strands are reverse complements of each other. So we know that there is a base pairing of G and C and A and T, but certain processes that go on will only be able to read. So if you have protein binding, they may only happen in one direction along the DNA strand. And that directionality of DNA proceeds, if it proceeds in this direction, 5' to 3' on one strand, it actually proceeds in the opposite direction on the opposing strand. So you may have something that could bind in the top strand, or it could also be binding to the other strand. It isn't picky, but in whichever strand it binds to, or whatever strand that process happens, it must take place in the same direction. So as a result, you get this opposing directionality of DNA because of the biochemistry. And so for this reason, we say that the reverse complement of a string, a DNA string, is to take the complementary nucleotides, but then also reverse them because they're going in the opposite direction. So we'd be tempted to say that the reverse complement of AGT, etc., is TCAGC, but we need to take its reverse as well for biochemical reasons. This gives us a problem. I give you a DNA string that's we call text, and you tell me what its reverse complement is. Another relatively simple problem because it's not an involved problem, but we have to range through a text and change some symbols and so on. And I always ask students to pause for a second, which is what I'm going to do now, and try and write the shortest possible pseudocode function that would solve this problem. So it's worth taking a minute or two. I'll take a quick break, and then I'll continue on. So often students will think, and there's a lot of different possible solutions for even something that's simply stated as reverse complementing a text, is that we could make one loop over a text and be producing a complementary strand by doing one for loop and checking kind of the opposite end of the text and creating a new symbol that is its complementary symbol. And so we would kind of be going from have a for loop that starts at the end of the text and goes backwards and produces a text that's the reverse complement of text. The way I would do this, though, would be to be very lazy. To say, why don't I just apply modularity? I'm going to assume that there's a function called reverse that reverses a string text. I'm going to assume that there's a function called complement that takes the complementary nucleotide of each position. It doesn't reverse it. And then if I want the reverse complement of a text, I can just reverse the string and call it X. I could call it text even. Call it X, and then complement every nucleotide and produce a string Y, and that's the reverse complement of the original text because I did the reversal and I did the complementarity. I don't even need to declare variables. I could just say take complement of text, apply reverse to it, and then return that. And then it's just a matter of writing each of those two subroutines, which I've kind of split my initial problem into two easier pieces. And I'm going to have two functions that are simpler, as well as probably easier to debug. I know it's a simple example, but even this simple example illustrates a very powerful paradigm of how we can solve problems that I'm going to say a lot more later when we start solving problems that are much more complicated, or at least seemingly complicated, that you might imagine would require these enormous functions, but that in practice we will have relatively short functions that just call each other very intelligently because we will design functions very well to communicate with each other. And that will allow us to solve more sophisticated problems with less stress than we have. And those types of programming paradigms are part of why I enjoy doing this, because they're the types of things that I see students often come in, even with experience, and struggle with because they don't necessarily understand that planning without sitting down and writing code at a laptop like Mr. Robot, but planning with pencil and paper and thinking about how to make our job easier on ourselves is actually very vital for solving problems often, and that we can solve sophisticated things with appropriate planning, and that little bit amount of time that we spend planning in addition is going to save us a lot of time down the road. I don't want to take too much of a detour to that, but I do want to point you towards some really fun things that we'll do very soon that apply this method. So maybe I found the hidden message here, and we look at k equal to 9 because now that we've learned about reverse complements, we realize that two of these 9mers are actually reverse complements of each other. And that's very surprising to find a 9mer that appears 6 or more times with reverse complements within a 500 nucleotide window. You could answer that question statistically. Even so, it's an involved process. And so I'll say also, we have these reverse complements because DNA doesn't really know which strand it binds to it. It doesn't have a preference of one strand versus another. Maybe the hidden message is ATGATCAAG. Maybe it's the reverse complement, CTTGATCAT. Nothing about this actually tells us which it is, because for each of those green strings that we see, we're only reading one of the strands. In the opposing strand, that's an occurrence of CTTGATCAT. So on the molecule, we have 6 occurrences of CTTGATCAT and 6 occurrences of the green string, 3 on each strand. And so nothing about this is indicated which of those two exactly that it binds to. We just know that they're surprisingly frequent. Then the next question I ask students is, now that we know the hidden message in the bacterium that causes cholera, or any other bacterium, how would we look for a hidden message that starts replication in some other bacterium? Hey, we sequenced a new bacteria or bacterial species. Here's one thing we could look for. Where is the where is the replication origin? So how would we look for that hidden message. And sometimes I ask simple questions, and students will say, well, this is a simple question for sure. Why don't I just look at other experimentally verified regions and verify that those two strings of length 9 are actually frequent words in those replication origins, and then maybe I build that out as an approach where I just need to search for those words, or at least clusters of those words in an entire bacterial genome. And then we apply it to some other species. Here it's T. petrophila, a bacterium that lives in oil wells. And we see that, well, actually there's 0 occurrences of those frequent words. If I apply the frequent words problem to that experimentally verified ORI, I get different 9mers. I get 6 of them, in fact. And so we see, now we're starting to see a more general theme of biology, which is that problems can become very difficult. It becomes difficult to extend something that we see and assume that it takes place in all different life forms or even similar life forms. Okay, so our conclusion here is that you might have different hidden messages for different genomes. The candidate hidden messages that we would find would be to say, well, actually in this case I see with reverse complements I find a string of length 9 that occurs 5 times. All right, and so maybe that's our candidate hidden message, but it's a different hidden message than what we saw before. And so I'm still stuck with the problem of needing an experimentally verified replication origin in order to find those candidate hidden messages. How can I find the replication origin to begin with? And that's problem 1. That's saying, yes, if you give me the short window, we can maybe find some hidden messages, but I still don't know how to find the replication origin in a much longer genome. I don't want to have to depend on needing that 500 nucleotide long window if I have 3 million base pairs that I'm considering. And I would ask you to think about, now that we know how these hidden messages might differ, how might we look for the replication origin to begin with if I give you a newly sequenced bacterial genome, several million nucleotides long? What is it that you would do to look for the replication origin if we don't have access to a lab where we're going to be able to run a bunch of knockout experiments and spend a huge amount of time to verify what window the replication origin is? So I'll pause there and allow you to think for a minute. So we have an algorithm that will find frequent words, that will look for, if I at least fix k, which here was equal to 9, if I fix k I can find the frequent k-mers. But that assumes that I know the region. So our old strategy was to look for these frequent words if I know the replication origin. Our new strategy will be kind of the inverse of that, which is that I want to look for the frequent words, and maybe that will tell me where the replication origin is. So I could perhaps look through all possible windows. And by windows I mean of length, say like 500 or however long that window length is, and look for frequent words in all of those windows. And if I don't see many frequent words, so whatever my maximum frequent k-mer is, it only occurs once or twice, maybe I throw that window out. And I'm going to need to consider all these overlapping windows too, because I don't want to kind of chop a region up that where we have a lot of frequent words here and it gets split over two windows. So I'm going to need to slide these overlapping windows along the string and say, well, where is it that I see windows that have frequent words in them? And maybe those windows are my candidate replication origin if I only saw one, for example. Here's where I want us to think back, and I'll pause again for just a minute and ask you to reflect on that strategy. Seems great, but before we jump into it, because we could interpret it in multiple ways, I want a computational problem. I want something that can be interpreted in only one way that will allow us to produce this strategy and say, I've solved this problem. But what exactly is that problem? What is the input and what is the output? So we will certainly need to know the genome for our input. That will need to be one of the parameters that we feed into any algorithm. If we write something that will find these clumps, clump finding, it's going to need to know the genome. But it will also need to depend on some things. For example, K will be an important parameter. How long of a substring is it that I'm looking for? Okay. How long will the window length need to be? If I'm writing something that's completely general, I'm solving a very general problem. Maybe this changes when I change, say, L equal 500 to L equals 700 or 1,000 or 200 or whatever it winds up being. And then if I'm really looking at what we did before, we threw out everything that didn't have at least some parameter T, occurrences of a frequent word. And so it can get quite involved quickly, this process of stating computational problems. And my sense is that, especially from checking out the chat, but seeing this with students, is that they'll produce very slightly different problems even from this same formulation. So we have a very clear scientific problem, we have a very clear kind of general approach, but we're actually not ready to implement it because we'll see some variation in what people produce is what it is that we want to do. And that's okay. Some of that variation is okay, but we should at least represent that variation in terms of the problem that we're solving so that we, before we even start writing code, can communicate about it. Okay? So I'll say that we're going to need a genome. We need K, L, and T. And what we're producing is the strings that produce these LT clumps. So K-mer is in an LT clump if there's some short window of the genome in which it appears at least T times. That's not the way that we have to solve this. We might take the starting indices of all the windows where we find an LT clump, for example. You might take only the window that achieves the maximum number of frequent words. So we would only produce one window. That might be dangerous, though, and we'll see why. So I can just present some pseudocode for doing this. It's an involved computational problem. It took me a second to describe. We have 4 parameters. And yet again, if we step back, we don't need to know anything about the syntax of the language. We can reason through how to solve this problem, especially leveraging the fact that we already know how to produce a frequency map. Remember, I can already produce, we'll implement this later, a function that takes a string and an integer k and produces that table of how many times every substring of length k occurs in that string. So I'm going to use that as a subroutine, and that will help me make this much shorter. So I could say, well, if I want to find these clumps, I'm going to need a list or an array of strings of length 0. At the end, I'm going to return that list, and so I'm just going to append to it every time I find something interesting. So I'm going to have n as the length of the text, and I will range over all possible windows. So all possible windows would be from 0 to n minus L. And so the current window is, well, if L is 500, it's that substring of length 500 of the genome, which I've called text here. But that substring starting at position I of length 500. So in general, it's text of I to I plus L. That's my current window. Within that window, I'm going to produce a table. So that's going to be that frequency map table that we've used before. And then I'm going to say, well, if I'm in a fixed window of length L or of length 500, as the case may be, if I'm in a fixed window and I've got that table, what are the things that occur more than or equal to T times? If I see something in that window of the genome that occurs at least T times, I'm going to append it to my list of patterns. And later I could remove duplicates. Or you could say, if it occurs at least T times and I haven't seen it thus far in the patterns, then I'm going to append it to my list. Either way of writing that. And then at the end, I've got a collection of strings. So an array of strings or a list of strings that is going to represent everything that occurred at least T times in some window. And look at how tight that function is, especially because, again, it leverages that idea we wrote before, that subroutine that we already have, our frequency map. And you're starting to see what I'm hoping to convey, which is that we don't need to solve this problem all at once. We can split it into subroutines and use these as building blocks for creating more complicated functions. And now, so I would always ask biologists alone why looking for these clumps in bacterial genomes is destined to fail before we ever try and implement it. We're welcome to write this function, and there's actually a hidden way in which we can make it faster that I could leave to you to think about. But I always ask people who are interested in biology why we know, before we even apply this, that looking for clumps is not going to work in order to find the replication origin. And the biologists will always tell me, well, if you're looking for something that occurs frequently in a short region of a genome, you're going to find a lot of that, because a lot of stuff is based off of binding, and it's not just initiation of replication. And having more copies of something is going to be beneficial in many different contexts. And genomes in practice will have many repeats. Some of them will be useful and some of them won't be. So the ALU sequence that we all have is about 300 base pairs long, and it occurs with some minor variations because you'll have some mutations 1 million times in each of our genomes. It's 10% of our genomes. It doesn't seem to serve a great purpose. It's even been potentially implicated in disease. And yet this is 10% of our genome. Is just this one repeat over and over again throughout the genome. If I write the code for something like E. coli and say, what is it that clumps together? What are the strings of length 9 that occur at least 3 times in a window of length 500? we could tweak this to find reverse complements as well, but we're going to find a lot of those too. And we'll have almost 2,000 of them. And it's completely unclear which one of those we should try. We don't want to have to do 1,900 different laboratory experiments. That's no easier than what we started with, where we're hacking out a DNA fragment and seeing if the genome can replicate without it. And it seems like maybe the computational folks retreat and they say biology is, okay, perhaps biology isn't boring, but it is just too challenging to try and apply computational approaches to. So lo and behold, a surprise is going to arrive for us, and that will be by stepping backward and doing an even simpler computational analysis, which is what happens if I simply look at the frequency of the 4 symbols. And I slide a window like we were doing before, but I'm simply counting the number of occurrences of A's, C's, G's, and T's in 100,000 nucleotide long windows of E. coli. That's something that has a verified replication origin. And all of a sudden we see, well, here's me counting the number of C's, and I've labeled in this plot where the replication origin ORI is and where the terminus is, TER. And it seems like between ORI and TER, you have a lot of C's there, and then as soon as you pass TER, you start not seeing C's. So you have fewer than what you would expect at random, which is 25% C's. And then let's count a different symbol. Let's count the G's. And the story is completely switched. So I slide the windows of length 100,000, and I count the frequencies of G's. So I take number of G's that have occurred over the total. And here, on one half the genome, I have less than 25%, and on the other half, I have more than 25%. And this looks significant because of how much, the pattern that is clearly evident, as well as the fact that it's, and for some of these windows, as much as a 2% discrepancy from what you might expect with flipping a coin or rolling a die that's 4-sided for picking nucleotides. And because I had 2 very different pictures for C's and G's, let's see what happens when I take the difference of the 2 frequencies for those windows. And there isn't a single window that we see that doesn't follow the same pattern on one side of the replication origin, and then on the other side it takes the opposite pattern. So we have, in many cases, over a 2% discrepancy between frequencies of G's and C's from 0, which is what we would expect for a random string. So genomes are certainly anything but random. This pattern is very stark and not immediately obvious why it would exist. It's still there, of course, even if I didn't know where the replication origin was and I just started counting at some random spot. So let's learn a little more about replication, dig back into biology, and realize that the more we understand about biology, we may actually have a very simple computational approach here for solving this. And that, again, is to come back to the directionality of DNA and say that you have DNA strands that run in opposing directions from 5' to 3'. I've got a blue strand on the outside that runs clockwise and a green strand on the inside that runs counter. I could use 4 DNA polymerases, which are the enzymes that facilitate copying new bases into an existing strand of DNA via the semi-conservative model. And this would work well with just 4 of these DNA polymerases proceeding outward in 2 directions on one strand and 2 directions on the other. And you would have this replication fork kind of, we're unzipping the DNA, and then we have new zippers being formed along the way. And we could do this with 4 enzymes and continue as this replication fork enlarges. This is the point where biologists are typically screaming at me because this just isn't right. The DNA polymerases are an example of one of these molecules that I mentioned, one of these enzymes that's unidirectional. It's only able to travel down a parent strand if it's traveling 3' to 5'. So what this means is that everything will proceed fine if I'm reading 3' to 5' away from the replication origin. So here it's going to be okay, and here it's going to be okay, but I actually have to read and add nucleotides in the opposite direction on the other 2 half strands, which means that I don't want to start this process of reading backwards too quickly. I'm just going to keep dead ending into something that we've done. I need to wait for a little bit. And so these half strands where everything proceeds naturally are often called forward half strands or leading half strands, will be the terminology that I use. And there's no problem replicating these, or we might call them reverse half strands, there's no problem replicating these leading half strands. Everything is proceeding normally and contiguously along those leading half strands that are shown with thick lines. There's a big problem, not a big problem, but we have to wait when we're replicating those lagging half strands, which are shown as thin lines. So if I'm adding nucleotides to either of those, I need to wait for a bit before I can proceed 3' to 5' to head back towards the replication origin and move in the legal direction. And I would add as a note, the leading and lagging half strands, note here that they're complementary. If I'm on a leading half strand, I'm always complementary to a lagging half strand and vice versa. So I have to wait until the replication fork opens, and then I have to replicate backwards, adding free-floating nucleotides backwards towards the replication origin. And then it's a matter of waiting a bit, and you'll have on the leading half strands everything proceeding as normal. And then we need to wait a little bit for the lagging half strands, and we're able to replicate backwards towards the replication origin. So those are called Okazaki fragments, and many of those fragments wind up being replicated, and another enzyme called DNA ligase has to then go in and sew them all together, and once that's done, the genome has been replicated. So this is all very nice, but what do we care? Well, note that you have two very different lives. You have the leading half strand that lives this double-stranded life most of the time, even during replication. As soon as the replication fork unzips, then you have DNA polymerase is fine to just slide in or continue adding free-floating nucleotides, and then that lagging half strand is sitting there single-stranded waiting to be replicated. Why would we care? Well, we care because single-stranded DNA and this asymmetry will be evident because the single-stranded DNA has a much higher mutation rate than double-stranded DNA. Some folks have proposed that this is why DNA exists, because the nucleotides themselves are locked against each other most of the time. So RNA might be a precursor to DNA because RNA is single-stranded, but it has higher mutation, and so by base pairing we produce a molecule that's more stable. If DNA lives single-stranded, then it's able to be mutated by different processes with a higher mutation rate. And so if one nucleotide, for example, for some reason had a higher mutation rate than the others, then you would probably see its shortage on the lagging half strand because it lives the single-stranded life more often. You're not going to see none of it because this isn't random nucleotides. It's things that have biological meaning, but you will see, presumably, more mutations on that lagging half strand. So the answer is deamination. There is one nucleotide that will be more prone to mutating, and that's cytosine, which through this process of deamination can mutate into thymine, and the mutation rate is 100 times higher on single-stranded DNA. So again, let's look at what would happen. You would have a lagging and a leading complementary half strand. Maybe you have a GC base pair there. And if you have a C on the lagging half strand, then on the leading half strand you bring in a complementary C and everything is fine. But on the lagging half strand, it's sitting there waiting for an Okazaki fragment to be produced. And so perhaps what happens is that you have that C deaminates into a T. And when the base pairing happens now, you won't base pair a G, which is the original base pair. You would base pair an A in that strand. So you have two, whereas through the replication process you started with one double-stranded DNA molecule. You now have two, and you have kind of one chromosome that has that correct CG pair, but another one which has TA. So you would have, for example, an organism that divided into two. You had a cellular division, and one of the children had a CG correct. It had inherited the CG pair, but the other one had an AT. And these types of things, these mutations could build up over time in a population. And so what we might see then is a shortage of C on this lagging half strand because it's more likely to mutate into a T. But also note that what happens in those organisms that pick up this C to T mutation, you're also going to increase the number of A's on the leading half strand, and you're going to lose a G in the organisms that have that mutation. So we'll have a shortage of C on the lagging half strand, but you'll also have a shortage of G on the leading half strand. And now we go back to those original figures that we saw, and we think we're really onto something. Because if we took this walk along the genome and we kind of were noticing that if we're on the leading half strand, well, then we should have a normal amount of C, and G should be low. So if we were looking at the count G minus C, because G is low and C is normal, and we were counting G's minus C's, we would see a decrease in that count of G minus C's. On the lagging half strand, that's where we're losing C's through deamination, but the G's aren't changing. And so as a result, we would have a normal amount of G, we'd have a low C. So if we took the difference in the frequencies of the G minus C, then that would be above 0. So we would see an increase if we were counting G's minus C. So again, if we're on the leading half strand, the numbers of G's that we see minus the number of C's, if we keep tabs, that should be going down. And then once we pass the replication origin and we go on to the lagging half strand, we should see that that count goes increasing again. So I would ask, well, what happens if we're walking along the genome and the number of G's minus the number of C's that we've been counting had been decreasing, but then on average it suddenly starts picking up again? Where are we? And you would note, well, we've passed from a leading half strand onto a lagging half strand, and so we must be at the replication origin. That's all well and good. But then we could say, well, I could give you a genome. So that's your input. What output is it that you're trying to produce? And I'll have you pause for a second. And again, reflect on what is the input? Well, it's probably a genome, but exactly what computational problem are we solving there? Because perhaps we can really find the replication origin, but I need to know exactly what output I need to produce. If you sum up the number of G's and keep track of how many G's you've seen and how many C's you've seen and take that difference, you could store this as you walked along the entire genome just in an array. So we could say some array skew, for example, for a given genome, would be to say, well, skew of K would be, let's count up the number of G's minus the number of C's for the first K nucleotides that we see. And I could even plot this. So once I have a skew array, I could plot every position K, I could plot the number of G's minus the number of C's that I've seen. So I would start at 0, and for this hypothetical string at the bottom, I would start with a C, so I go to -1. A and T don't affect this. Every time I see a G, it will go up 1 because I've seen an additional G, so I go 1 back to 0, another G back up to 1, and then another G up to 2. Then I see a C, so it goes to -1, and so on. Okay, so I can keep track of this. And then the question becomes, what is it that this skew array of a bacterial genome is going to look like? If I know that on this lagging half strand, or leading half strand, that the picture of decreasing versus increasing is going to be different. What's going to happen if I plot this skew array? And I know that for half the genome, the count of G minus C is going down. And then for the other half, if I'm counting the number of G minus the number of C, I'm going to be seeing more G than C. So it's going to be tending to be more positive. What is it that that skew array is going to look like? And here many students will say, well, I bet it couldn't ever possibly be so perfect as this, because we have seen a little bit of a signal. We've seen a pattern in our data. But on the whole, I'm sure that it might be difficult to even visualize, but probably if the number of G's minus the number of C's is decreasing on one half strand, we would see a decrease downward. And then for the other half strand, if I'm keeping track of this, it might turn around and start heading back upward. But no one can really, who hasn't seen this plot, can believe for a messy biological data set like the genome of E. coli, what the skew diagram is really going to look like and how perfect it is. That when you plot the position of a genome against just a simple count of the number of G's minus the number of C's that you've seen up to that point, you get this perfect V shape, practically. And it's a little off from centered because the genome is circular. So we're going to come back, the pattern is continuing there at the end and we'll meet the origin. Imagine kind of wrapping this plot around on itself. But remember, I said before, if I were walking along a genome, so just reading through a genome file and seeing that if I read through this genome file, I was seeing that the number of G's minus the number of C's was really decreasing a lot. And then all of a sudden it started increasing again. Where were we? And that's the same question I asked before. And we said, well, we've got to be at the replication origin. We're switching from one half strand to the other. And so there's the replication origin of E. coli. There's the experimentally verified one. And as a result, we have now solved question 1. We don't need to run knockout experiments on any sequenced bacterial genome in order to figure out where its origin of replication is. Here is kind of the field of computational biology in a nutshell, that we can save our time of not needing to run very costly, both in terms of time and in terms of funding, experiments for many hundreds or thousands of bacterial species. We can simply look at the minimum region of this genome for anything that has a sequenced genome. And maybe there are many other questions that we can answer too once we have a bacterial genome, not just what the origin of replication is, but what is it that the bacterium does? How is it that we can compare it to other bacteria, right? That sort of thing. And so we've solved problem 1. Remember we said that question 2, which was if I give you the replication origin, what's the hidden message? We worked with the frequent words problem. And so for each of these 2 questions that I had, we can solve the appropriate question. For the question 1, which is where the replication origin is, I give you a genome, and you give me perhaps the min value of this skew array for the genome. That was a research paper that came out as bacterial genomes started to be published in the mid to late 90s. You saw this paper is from Grigoriev, and it has several hundred citations. So it's the easiest or the simplest imaginable type of analysis that we could have done on a bacterial genome to produce this beautiful skew diagram. Right? What an amazing time it would have been to be working with this data set, because the simplest analysis you could have applied resulted in a paper with several hundred citations. Don't hesitate, don't think for a second that there aren't many other different types of really cool data sets that are just now being generated in biology or whatever cutting edge discipline that can provide some really cool insights without necessarily needing the most complicated imaginable type of analysis. So that was a research result from a couple decades ago. And I would point out that, yes, nothing in biology is ever going to be perfect. If I look at E. coli, I'll actually find that there are no frequent 9-mers that appear 3 or more times in this experimentally verified replication origin, and the one that we find when we apply our skew array approach. And we say, should we give up again? And so here we think, well, maybe there's some biological process that we can reason through, and we've already seen that mutations are just a fact of life in the world of DNA. And so if we wanted to incorporate that, maybe we look for frequent strings that account for not only reverse complements but also mismatches. And when we do that, we find here's a string that occurs, a string of length 9 that occurs 4 times with reverse complements if we allow one mismatch. Of course, there are going to be more complications than that too. For many bacterial genomes that you for many bacterial genomes that you sequence, you'll get a very pretty skew diagram. There are some that don't provide a nice skew diagram, but many do. I think most, in fact, produce a nice skew plot. And it's always pretty to see them because you can say, well, there's its replication origin, kind of screaming out at me. Here's one for that T. petrophila that I mentioned, where the skew diagram is not quite so pretty. We can see that perhaps maybe because it's flattened out on one half strand and maybe steep on the other, maybe at that kind of turning point is where the replication origin is, but it's certainly not something that we would have been as confident about as we were for the skew plot of E. coli. Some of these bacteria will have fewer DNA boxes like we saw with E. coli, and you have these complicated drawings, and also you may not have a terminus that's exactly opposite from the replication origin. Is there a moral, though? I would say certainly there is, which is that this type of division of sciences is, well, you have biologists over here and computer scientists over here, and there's no value in getting them to speak to each other, and we should just propagate the stereotypes that might exist of those two disciplines is not an appropriate view of how biology or science in general can and should operate in the 21st century. And so this is a great microcosm for why my own discipline of computational biology exists and is flourishing today, because there are so many problems that are like this, where you need an understanding of both fields in order to truly attain some cool results. So that's it for today. Next time, I'll use this material in order to motivate learning and working with strings and maps. And even though this is biological stuff, the algorithms themselves are very natural to do in an introductory context when working with strings and maps. So that's what I'll do. Before I leave you, I'll say that I always give special thanks to Pavel Pevsner, who I worked on this material with initially as a PhD student, and has been kind of my partner in crime in producing online educational materials in computational biology over the last few years. Developing this story was always thanks to him, and it's always been fun to teach. So until next time, I thank you for your attention, and I look forward to seeing you then.